Hello and welcome back to your Chem 4 lecture. We're finishing chapter 16 today with a discussion on enzymes, a particular class of protein that acts as a biological catalyst. So of course, all that we talked about, we build up these proteins from the primary structure to the secondary to the tertiary to the quaternary. Now, what do they actually do? And one of the most important ways that they act in our bodies is that they catalyze basically every biological reaction that occurs in our body. So yeah, that's what we're going to talk about here. Okay, so real quick, let's go ahead and review what the meaning of the word catalyst is. Basically, catalysts are a class of compounds that can speed up a reaction. So this can be as simple as, for example, with hydrogenation of alkenes. Remember, use platinum, nickel, or something like that. And all it does is it changes the activation energy of a reaction. So for a normal reaction, for example, carbon dioxide reacting with water to produce bicarbonate, activation energy without an enzyme is very high. So this reaction doesn't. However, if you instead catalyze the reaction with an enzyme, the activation energy lowers. So you only need a small input of energy to actually get the reaction to occur. And this happens across the board. Virtually every reaction that occurs in our bodies takes advantage of an enzyme that does it. Yeah, again, so enzymes catalyze almost all the reactions that occur in the body, and they increase the reaction rate by lowering the energy of activation. So for example, the one we were just talking about that takes carbon dioxide and turns it into bicarbonate and a proton is a carbonic anhydrase, for example. So note that name there. A lot of enzymes tend to end with the suffix ase, and basically what it does is to name an enzyme, you're going to name it based on the reaction that it catalyzes. So you take the name of the reaction or whatever compound it reacts with and add in the suffix ase. So for example, if you're taking sucrose and you're splitting it in two, remember sucrose is a disaccharide. It can be split into its monosaccharides. Well, you would need an enzyme to catalyze that reaction. That enzyme is sucrase. Okay, how about that? Next up, there's a series of oxidation reactions in the body that we're going to talk about in the future. Oxidase is a class of enzyme that catalyzes oxidation reactions. And then sometimes we have some common names for digestion enzymes, because these have been around for a long time. We've learned about them for a long time. So we have pepsin and trypsin as names for enzymes that basically digest various compounds in our metabolism. Okay, so here's our classification of enzymes. We start from oxidoreductases, which of course catalyze oxidation reduction reactions. We have transferases, which transfer groups of atoms from one location to another. We have hydrolases that catalyze hydrolysis reactions. There are lyases. These in particular add or remove atoms to or from a double bond. So I know that sounds confusing, and it does seem pretty confusing, but remember, you're either going to add an atom to a double bond or remove an atom from a double bond. Isomerases isomerize compounds. In other words, they rearrange the atoms in a compound. And then finally, ligases use adenosine triphosphate, that's ATP, to combine small molecules into bigger molecules. Okay, so given that, these, of course, you just need to learn the definitions for. Go ahead, review your notes, and then when you're ready, start sample exercise 10 here, where you're going to match the type of reaction that's catalyzed with its enzyme class. We have 4 of them listed: hydrolases, isomerases, transferases, and ligases, and then match it A through D. So pause the video, review your notes, and when you're done, let's take a look at the answer. So pause. All right, let's take a look. Okay, so what type of atom or what type of reaction is catalyzed by which enzyme class? Form bonds between molecules using ATP energy. That ATP is a giveaway there. That means it's a ligase. Rearrange atoms in a molecule to form an isomer, isomerase. About transfer a group between two compounds, that's a transferase. And then finally, hydrolysis reactions, those are hydrolases. Okay, so now how do enzymes actually do their catalysis? Well, I'll do my best to explain it to you here, but I just want to point out this is some of the most complicated chemistry that exists possibly in the universe, I would say. The biological systems that make up everyday biochemical reactions are intricate, and they involve a lot of different interactions in a protein. And just as a chemist, I find it really fascinating because it seems just almost impossible that they do the things that they do. But we're going to explain it to you as well as I can. Okay, so the way we're going to start is by exploring the structure of the enzyme. And on the surface of an enzyme, there's going to be a small region that's actually responsible for where the reaction takes place, and we call that an active site. The active site will bind a substrate, and then some reaction is going to occur with that substrate. Substrate is just a reactant. Okay, so this word active site, again, it's a region on an enzyme, and it fits whatever shape of the reacting molecule is going to be, and that's what's called a substrate. Okay, the amino acid R groups are going to bind the substrate. So if you have a substrate that's nonpolar, you'd want nonpolar R groups to help cushion it. And if you have a polar substrate, then of course you'd want the polar or hydrogen bonding R groups as well. And also you need to make sure that the active site is able to release the products when the reaction is complete so that it can do it again. Remember, if you had such a good binding site, so imagine that the active site really liked the substrate, well, the substrate is just going to bind to the active site and then it'll stay there, right? So there needs to be some inducement to do the reaction and then to actually have the reaction occur. Okay, so in an enzyme catalyzed reaction, what happens is the enzyme will have a substrate attached to the active site. This is forming what's called the enzyme substrate complex, or ES. So this should be an arrow, sorry, E plus S goes to ES. Now we have our enzyme substrate complex. Now in this potentially momentary complex, we have a reaction occur, and then products are going to be released from the active site. That opens up the active site to take in another substrate and again do the reaction and release the products again. So the general form here is that an enzyme reacts with a substrate, forming an enzyme substrate complex, and then products are released. So you have the enzyme available again and the products. And yeah, so for example here, if you have your substrate here, you could form example like salt bridges here to from your substrate to your active site. You could form hydrogen bonds. You could have hydrophobic interactions as well. All of those are going to be involved in this. Okay, so let's look at a particular one. So if we have a disaccharide, so starting from lactose, we know that lactose is a disaccharide, and that means that it can be split into its two monosaccharides. So what happens is the enzyme that's responsible for breaking up lactose is called lactase. So lactase reacts with lactose, forms the lactase-lactose complex that then breaks up the lactose into its components, the products glucose and galactose. The products don't have an affinity for the active site in the same way that lactose does. So the products are released, and then the lactase is able to do it again. The way that lactose fits into the active site is such that the glycosidic bond of lactose is in a perfect position for a hydrolysis reaction to occur. So R groups on both sides of on all sides of that, both on the glucose and the galactose side, are going to be kind of like pulling them apart a little bit and then letting that hydrolysis actually occur. And these product structures are no longer perfect for the active site, and so they're released and another lactose actually comes in. Okay, again, I'm describing it in very general terms because the actual interactions that occur are going to be specific for any particular type of enzyme. So we just have these kind of like really nice pockets where the substrate fits, but not perfect because it's going to be teased into a position where it wants to react. The reaction is going to occur, and then all of a sudden it no longer fits and it floats out as the products. Okay, so we have this kind of older model of enzyme action called the lock and key model. We don't really use this anymore because it doesn't really understand. Basically, it's like, okay, the enzyme is the lock, and the, wait, was it? Sorry, this is such a bad one that we don't even really use it here. So the lock and key model is an old model. And basically it said that the active site was a rigid, non-flexible shape, and the substrates only binded an exact fit like a lock. So this is bad. Let's just not talk about the lock and key model at all. Instead, we want to talk about the induced fit model. We know that these enzyme structures are flexible because there's a little bit of give with any hydrogen bond-based structure. So, alpha helixes, we draw them as rigid structures, but those can flex and bend, and there's a range of distances that those hydrogen bonds can take up. And an enzyme structure is flexible, and it can adjust to the active site in order to bind to substrate. So the range of substrate specificity is going to increase. In other words, you can have substrates that are exactly the substrate that the enzyme has evolved for, but you can also have very similar mimics that kind of fit in the active site and can clog up the works that we'll talk about. And then we can also explain why the reaction actually occurs and why it is that the products leave, right? Like a lock and key, you do the lock and key and then you pull the key out. The key wouldn't leave on its own. But if we understand having the fluidity of the active site, then we can understand that the active site is going to kind of push the products out at the end. Okay, so this is an example of the induced fit model. So going back to lactase and lactose. So what happens is the substrate and the enzyme work together to give a particular fit, so a particular arrangement. And what that's going to do is allow the reaction to actually occur in a way that's just a little bit easier than it would without. So like lactase, for example, the way this works is the lactose will sit in the active site, and it positions the glycosidic oxygen here in such a position that just a little bit of pressure is kind of pulling on the two monosaccharides apart, and that's going to lead this oxygen to be more reactive in the middle. So that's the induced fit. It's when we put the substrate into the active site that changes the shape of the active site. And now you have your enzyme substrate complex is going to be a little bit different in shape because it's got two things adding up to a third thing. Then the reaction actually occurs and you're back to normal enzyme that's going to have the induced fit here once again. Okay, so so that brings us to certain changes that we can make to enzymes to study how the activity actually works. So let's go ahead and talk about isoenzymes. So isoenzymes are, you might think, oh, it's iso like isomer, an enzyme like what we've been talking about, and that's the idea. It's a different form of an enzyme that catalyzes the same reaction in different cells or different tissues in the body. And basically what happens is the quaternary structures in these isoenzymes are going to be slightly different based on very small variations in the amino acids in the primary structure. So for example, lactate dehydrogenase catalyzes the conversion between lactate and pyruvate. These are the monomers listed here, lactate and pyruvate. Sorry, monosaccharides here. And those 5 differences can occur in different places. So each lactate dehydrogenase, thank you, each lactate dehydrogenase isoenzyme has different polypeptide subunits. So there's 2 polypeptide subunits, M and H. Different combinations of M and H are used in different organs. So in the liver and muscle, lactate's converted to pyruvate by LDH5, and that has 4 M subunits. However, in the heart, you have 4 H subunits actually doing it there. And you can imagine, liver and muscles have a different structure than heart. And so there's going to be, differences needed based on what type of, what the surrounding of the enzyme is actually going to be. So if you have a myocardial infection, you can actually, it can be seen if you notice that there's lactate dehydrogenase that should be in the heart, but is instead in the blood flow. So if you have a myocardial infarction, that's a heart attack. If it damages the heart muscle, some of this H4 LDH is actually leaked out into the blood serum. So if you detect it there, that's a pretty good indication. On the other hand, if you notice LDH5, which has 4 of the muscle subunits there, M4, that's an indication that you have some other possibly liver damage or other disease. So here's the different LDHs. So LDH1 we mentioned has all 4 H's. LDH2, which is present in the red blood cells, heart, kidney, brain, has 3 of the H's and 1 of the M's. And then brain, lung, white blood cells are H2M2. Lung and skeletal muscle are HM3. And then finally, skeletal muscles and liver have the M4. Okay, so let's go ahead and take a quick quiz on the this idea of enzyme here. So pause the video here, answer these questions by checking your notes, and then when we come back, we'll have the answer available to you. Okay, so sample exercise 11. The active site is A, the enzyme, B, a section of the enzyme, or C, the substrate. It is a section of the enzyme. And then finally, in the induced fit model, the shape of the enzyme when the substrate binds, it adapts to the shape of the substrate. In other words, they both kind of modify in order to accommodate each other. Okay, next up, factors affecting enzyme activity. So you can shut down enzyme activity, or you can speed up enzyme activity by using just a few different factors that can change it. Okay, so enzymes have an optimum temperature that they act at. This makes sense. For example, like in our blood and in our body, our bodies are maintained at temperature right at 37 degrees Celsius. So it makes sense that the proteins that are in our body, our enzymes, should operate very well at 37 degrees Celsius. That's what they've evolved for. So and that's exactly what we see. Because these enzymes are shape dependent, and also those shapes, as we said, are not based on chemical bonds. They're actually based on very relatively weak interactions like hydrogen interactions, hydrophobic interactions. Your enzymes are only going to operate at a very narrow range of temperatures. At low temperatures, they're not going to have the right shape, and molecules aren't going to be moving fast enough for the actual enzyme to catalyze the reaction. And then also above 50 degrees Celsius, well, that's when you actually denature proteins. And so you lose that shape permanently. And you see no catalytic activity once it's heated over 50 degrees Celsius. Now, there's temperature, and the other thing that modifies it is pH. Now, we've seen already that R groups sometimes have acidic side chains or basic side chains. And as you can imagine, something that's acidic, if you put it in a base, the acid's going to react, and so you're going to lose the same structure that kind of holds things apart, that holds things together. So our enzymes operate at an optimum pH, and that pH can change based on the location of the enzyme. For example, there's certain enzymes that need to react in our stomach acid, and their optimum pH is very low, like something like 2, for example. But in our blood, our pH is around 7.4, and sure enough, that's exactly where our maximum enzyme activity is taking place. So yeah, so enzymes in the body have an optimum pH of around 7.4, but again, certain organs can be different. So pepsin, this is what I mentioned in the stomach, pepsin catalyzes the breaking apart of peptide bonds, and it's best interacting at an acidic, very acidic environment of 1.5 to 2.0 for pH. Now, lactase, on the other hand, is in the GI tract, gastrointestinal tract, and that's where we're going to take apart lactose, and its optimum pH is around 6. Again, it just kind of fits in with the location where it has been, where it's evolved. You don't need to memorize these here necessarily, but just know that there's a range. So for example, in the pancreas, that's where we're going to tear apart these lipids and the ester bonds in them. And the optimum pH there for lipase is 8.0. And then finally, arginase, which eats up arginine, has an optimum pH of around 9.7 in the liver. Okay, so here we go. Sucrase, but this is a sample exercise, so make sure you're going to read this and try to come up with the answer. Sucrase has an optimum temperature of 37 degrees Celsius and an optimum pH of 6.2. Determine the impact of each change on the enzyme activity. And we're going to say no change, increases, or decreases. What happens if you change the pH to 4? And what happens if you run the reaction at 70 degrees Celsius? So pause the video, and when you come back, we'll take a look at the answer. Okay, well, no surprise here. Both of these is going to decrease the activity of sucrase. Again, we mentioned that these enzymes are very sensitive to both pH and temperature. So if we change the pH drastically by a whole 2 units of pH, we're going to decrease the activity of the enzyme. Likewise, running in a reaction above 50 degrees Celsius would completely denature the enzyme. And so it's going to decrease the activity of the enzyme down to 0. Okay, so that brings up our final topic here, which is the inhibition of enzyme. So not only can we denature enzymes and stop their activity that way, they can also have a problem where they run into an inhibitor. And an inhibitor is a molecule that causes a loss of catalytic activity. Basically, what happens is it's going to compete, or I shouldn't say that yet. We're going to talk about 2 different types of inhibitors. We essentially have an enzyme that can pick up an inhibitor and form an enzyme inhibitor complex that no longer is able to catalyze the reaction. So that's basically the competition is the enzyme going to react with the substrate, or is it going to react with the inhibitor? If it reacts with the inhibitor, then it's not going to react with the substrate. Okay, now I mentioned already there's 2 types of inhibition. So we're going to talk about the competitive inhibitors and the non-competitive inhibitors. So a competitive inhibitor is called that because it competes for the same active site as the substrate. So they tend to have structures that are similar to the substrate, and so they compete again for the exact same site. Now, the other good thing about a competitive inhibitor is that it is reversibly binding. So the enzyme can react with the inhibitor to form the enzyme inhibitor complex, but then it can also go back to form an enzyme, which can then react with the substrate and form the enzyme substrate complex. So basically, times when you have a high concentration of substrate, the enzyme substrate complex is still likely to form and form the products. However, if you have a high concentration of inhibitor instead you're likely to form the enzyme inhibitor complex and not form any product. So this is the basic example here, that if you take the same enzyme with the inhibitor, there's a competition for the same site, the active site. So if you form the enzyme inhibitor complex, you're no longer going to form the product. Only when the enzyme reacts with the substrate are you going to form it. Okay, so that's competitive inhibition because they're competing for the same active site. Now, on the other hand, we also have non-competitive inhibitor. The structure of the non-competitive inhibitor is usually very different from that of the substrate. Basically, what happens is the non-competitive inhibitor will bind to the enzyme at a different site, away from the active site. And what that does is it distorts the shape of the enzyme and permanently shuts down the active site. So the active site shape gets altered, and then no longer do you have that induced fit of the substrate into the active site. And again, this one, you can't reverse the effect by adding more substrate. So here what happens is you have a different shape, and this inhibitor just changes the shape of the active site so that the substrate no longer fits. And now all of a sudden you can't form the product down here. Okay, now I mentioned this already, irreversible versus reversible. Now, if your inhibitor binds permanently to the enzyme and permanently causes a shape change in the active site, or permanently bonds with the active site, then that's what we call irreversible. There's no way to remove that. So an irreversible inhibitor is a molecule that causes the enzyme to lose all activity. So this is usually going to be something, if it's an enzyme that your body needs, then this is going to be a toxic substance. It's going to stop that enzyme from carrying forward. So what happens usually is there's some sort of covalent bond that forms with an amino acid, and that prevents catalytic activity. So penicillin, for example, is an antibiotic, And this permanently, oh, I don't really have it here, but what this does is it permanently can react with several different R groups that permanently shut down bacterial activity. Nerve gases do the same thing, and insecticides kind of have the same goal, shutting down some key enzyme or some key biological process by shutting down whatever enzyme it's going to attack. Okay, so here's the things this is the summary of what you need to know. So if you have a competitive inhibitor, it's going to have a similar shape to the substrate. If you have a non-competitive, it's not going to be similar. And if it's irreversible, again, that's not going to have a similar shape to the substrate. It's going to have some covalent bond forming. Now, as far as binding away to the enzyme, the competitive inhibitor competes for and binds at the active site. The non-competitive inhibitor binds away from the active site and changes the shape of the enzyme. And the irreversible inhibitor forms some covalent bond with the enzyme that, again, permanently changes the shape of the enzyme. As far as reversing it, competitive inhibition can be reversed by adding more substrate. Non-competitive inhibition can be reversed by removing the inhibitor. And then finally, irreversible cannot be reversed, exactly what it sounds like. Okay, so that brings up sample exercise 13. Given the following descriptions, identify each of them as either a competitive or non-competitive inhibitor. So pause the video here, come up with your answers, and we'll take a look at the end of in just a moment. So pause. Okay, so let's go ahead and check it out. So A, increasing substrate reverses inhibition. Well, that sounds like a competitive inhibitor to me. It binds to the enzyme surface, but not to the active site. That's a non-competitive inhibitor. Its structure is similar to that of the substrate. That sounds like a competitive inhibitor. And then finally, inhibition not reversed by adding more substrate. That's non-competitive inhibitor. Okay, all right, that covers us for the chapter. I'll talk to you very soon on the next chapter about nucleic acids. Until then, let me know in the comments if you have any questions. Bye-bye.